In this video, we will tackle the topic of gene interactions. Gene interactions occur when two or more different genes influence the appearance of a single trait. This actually describes many of the traits we observe, which may also act in concert with environmental factors. However, we also have some simpler instances where two genes influence the phenotype of a single trait, and we will explore those here. Often, the best way to understand what we are exploring here is to look at an example. Here is an example of two true breeding white parents. When crossed to form a dihybrid, the offspring are all purple. This is what is known as a complementation interaction, where the wild type allele of both genes is needed to produce the purple phenotype. But what might be the molecular mechanism for this? Often, genes that interact are part of a single pathway that produces a particular protein. So, in our example here, you need the wild type allele for both enzyme C and enzyme P, or you get a colorless flower, either the precursor or intermediate. But what happens when you allow these purple flowers, these purple dihybrid flowers, to self-fertilize? This is where things get interesting. Before we jump into those results, let's think about what we would expect based on the rules of Mendelian inheritance. Recall that a dihybrid cross, which is what we are proposing here, should yield a phenotypic ratio of 9 to 3 to 3 to 1. However, in this epistatic interaction, we get a modified ratio for a dihybrid cross. Since a wild type allele for both genes is required to get a purple flower and any other combination yields a white flower, our ratio for this dihybrid cross is 9 purple to 7 white flowers. We will often look at these modified dihybrid ratios to ascertain the type of gene interaction in a particular cross. Because there are more types of gene interactions than just what we've seen so far, and a bit of terminology that goes along with it all. So take a moment with this slide to understand the terminology. I have also listed a few types of gene interactions or epistasis, and you can see how each is a modification of the 9 to 3 to 3 to 1 ratio. We will work our way through several examples in order to better understand this. A final variation in gene interactions for this video is gene redundancy. This happens when the loss of function in one gene does not affect the phenotype, but loss of function in both genes will affect the phenotype. As you might imagine, this arises from things like gene duplications, where these genes maintain very similar functions. Here is a quick example of this type of interaction, with a seed capsule shape that is normally triangular. There is a double recessive, which is ovate rather than triangular. However, our dihybrid cross in the F1 generation did not yield a 9 to 3 to 3 to 1, but instead a 15 to 1 ratio. This indicates that the ovate phenotype only occurs when both genes are homozygous recessive, and any other allele combination yields a triangular seed.